Hello, anatomists. Okay, we have learned a lot about the lymphatic system, and now we're going to dive into immunity, and I'm just going to make you this video. I think it's going to be easier if I do it in the video. A little bit of review. So, innate immunity versus adaptive immunity. Innate immunity is right out of the gate. It's what we're doing, boom, right there. We're starting out with a local effect right at the point of invasion. Remember, that's going to be our fever, that's going to be our swelling, that's going to be our non-specific defenses. It does not remember anything. It is innate immunity. You are born with this. Adaptive immunity, we are going to talk about, okay? It is defense against very specific pathogens. Okay, again, your first line of defense. What is your first line of defense? Your skin, your mucus, your membranes, everything that's going to try and, like, the outer layer, if you will. The second layer, this is going to be, let's say it makes it in through the nostrils. Right now you've got leukocytes, you've got macrophages that are going to bite it. You've got antimicrobial proteins, you've got natural killer cells, you've got fever, you've got inflammation. That is your second line of defense. Your third line of defense is going to be adaptive immunity. Okay, we've talked about the skin. This is just a review, right? You have keratin. It's not dry in a bad way, but it's dry so that stuff can't live on it, right? It's dry enough that you don't get bacterial living on it. You shed it very often, and you have some peptides in your skin and your mucus that kill things, right? This is review. We talked about this in class. You have membranes that digest things. You have sticky mucus in your nose. You have cilia in your throat that cough up. You've got lysosomes that destroy bacterial cells. This is tears in your saliva. What are we not going to do? We're not going to lick people. We're not going to lick wounds. We're not going to let dogs lick our wounds. That's not what that means. We have some viscous barriers, difficult through sticky gels. Again, mucousy. We have pathogens in those. Okay, second line of defense. Okay, so we've made it through the mucus. We've made it through the skin. We've made it through the mucus. We're inside now. Now we're going to get hit with some white blood cells. You got some leukocytes here, okay? We talked about these in class. Again, we just have neutrophils, eosinophils, basophils, lymphocytes, which we're going to talk about today. These are your T cells and your B cells and your natural killer cells. That's all part of this adaptive immunity. We will get to that. Remember, everybody's going, so if you come up here, lymphocyte, we're going to go to lymphocyte school. We're doing that when we talk about adaptive immunity. Right now, we're talking also acquired immunity. We're going to have B cells and T cells. Okay? We are going to have B lymphocytes. That's called humoral immunity. Okay? Humoral immunity, and that is your B lymphocytes. Both of these things are born in your bone marrow. Say that several times fast. Born in your bone marrow. They go to school in two different places. Okay? Your humoral B lymphocytes are going to go to school in your bone marrow. They have antibodies on them. I tried to draw this beforehand because it was taking me too long to draw it with my mouse. Right? Your receptors are antibodies. Okay? Now, your cell-mediated, this is your T lymphocyte. Again, it's born in the bone marrow, but it goes to school, gets educated in the thymus, and it has a receptor. Not an antibody, but a receptor. Okay? Those are some main differences between those two things. Also, if you remember, this is again just some review. Your MHC platform is like a Facebook page. So you're going to eat something. You're going to eat a thing, right? You're going to eat a pathogen. You're going to eat it. You're going to digest it. The Golgi and the endoplasmic reticulum are going to cut it up and send it out. And you're going to put it on this MHC platform. And you're going to be like, hey, I ate this thing. Okay? We're going to name this Bob, Jill, Joe, Sal. Let's go with Joe. Okay? This is Joe. We're going to come back to Joe. We're going to see what happens when Joe posts this. Okay. Some other review. Interferons, group of proteins. These are cytokines. They are secreted by virally infected cells. So once you get infected, you're going to sort of send out an interferon, and it's going to interfere with viral replication. That's one of the ways that your body has an antiviral protein, okay? We talked about this a little bit. Let's talk about complement. Actually, let's do mine first, okay? Because I am great at drawing. Let's move this up here. Yeah, excellent, right? So good at the art. Should have been an art major. You have complement. So what you have here, this is an antibody, right? And it has a little space for a receptor right here, right? And its little Y, like YMCA, right? It has a little receptor right here. When a pathogen binds to that, cascade of chemical reactions. We're not going to learn those. Nobody has time for that chemistry-wise. You take biochem if you want to know what all of these are. Take pathophysiology. You will learn them. What happens in this cascade of chemical reactions from complement? That's where your inflammation comes from. Okay? How did it get there? Antibody receptor, right? Starts the process. Inflammation, opsonin. What does opsonin do? It basically initiates a fever. Remember, we talked about fever. Ultimately, that gets you a MAC attack, membrane attack complex. Basically, what that does, it's going to pop you if you're a cell. It's going to come in and poke holes. It's going to pop you. Okay? Let's look at that in a more constructive way that is not me drawing it. It's part of the innate immune system, okay? So it's not acquired. This is just going to happen, okay? Activated in response to the presence of a pathogen or injured tissue, all of a sudden that antibody that we saw is going to bind, right, to the receptor, and through inflammatory response, it's going to attack the pathogen. First thing it's going to do, opsonization. Complement proteins coat the pathogen, damage it, and phagocytose it. What is phagocytose it? It's this. It's going to eat it like Pac-Man. Phagocytose it. Okay? Totally the same as my picture. That's what you were going to say. Also, totally the same, right? Does this not look like exactly what I just drew? You have your antibody, right? And your antigen. You have this whole thing. What does that do? It enacts a complement activation. Your antigen binds to your antibody. It starts complement. A whole bunch of things happen, and you get a MAC attack. What does that do? Pokes a big old hole. Cell lyse. Oh, let me move this down a little bit. Cell lyse, death. This is death. It pops holes in it. Okay? Looks exactly like this. Just saying. Okay. The system, the complement cascade. It is called the complement cascade. Okay? Sorry, I'm going to move myself around so you can see it. Inflammation, we already know that. Cell lysis, basically, it's going to MAC attack it. It's going to pop it. It's going to kill it. And then we're also going to have an immune complex clearance. This is going to remove all of that and all of that immune system from your bloodstream. It's going to get it out. It's going to clean everything. Okay? Again, this is complement. This happens when your antibody reacts. Here it is again. This looks very just like my picture. I know it doesn't, people. I know I can't. I know I can't draw. Here's your opsonization, tags pathogens for removal. Right, here's your big old macrophage about to Pac-Man that thing. Inflammation, right? A histamine, mast cells. It's going to just inflame and start an oozy, right, getting it out. Here's your pathogen lysing. Look at that. It's an action photo of a cell exploding from being lysed. And this is the complement cascade system. Here's another pathway. If another way, there's a classical antibody-dependent antigen-antibody complex comes together, right? It has on its little Y, it has its little receptor here, and it binds, starts the complement cascade, goes here. There's an alternate pathway with some other things that can bind. There's a lectin pathway that also binds. All of this is complement. I don't need you to know the C3s. Here's, again, more C3s. Again, what does it get you? It gets you inflammation. It gets you immune clearance. It gets you phagocytosis, eating it, right? All these things, it gets you. That's what I want you to know. You don't need to know about the C3A and the C3B. Natural killer cells. Delightful name. Love it. What are these guys doing? These natural killer cells are just out searching for something to eat, right? Continually patrolling your body looking for pathogens and diseased host cells. They're just out there like natural killers. They were born to kill. They were born to attack and destroy. Attack and destroy microbes, transplanted cells, cells infected with viruses, and cancer cells. And that's what they do. Basically, they release perforins, perforated, right? They're going to poke holes in you. See this? They're going to poke a little hole in you. They're going to insert granzymes. Granzymes enter from the perforin hole, and they just degrade you from the inside out. And the enemy cell dies by apoptosis. It just pops. And then the macrophage engulfs and digests the dying cell. This is a traumatic process, right? Remember, you're going to get fever, pyrexia, which is an abnormal elevation of the body temperature. It's from trauma, infections, drug interactions, brain tumors. Febrile is pertaining to fever. These are just vocabulary words. It's a defense mechanism in moderation, more good than harm, right? It promotes interferon activity, so it moves all that cascade around. It elevates the metabolic rate and accelerates tissue repair. It inhibits the reproduction of bacteria and viruses above 98.6. Remember, I said your bacteria, they don't like it above 98.6. We talked about this in class. Somebody even asked this question. Recovery is sometimes faster when a fever runs its course versus taking antipyretics, which are acetaminophen, ibuprofen, acetylsalic, any of those things. Sometimes. There's a point, right? Once you're like 102, 103, you got to get that down. 101, 100, you can probably just push through a little bit and see if it doesn't increase. You're actually going to feel better. It might actually, you might recover faster. So there's that. There are two types of fever triggers. Exogenous pyrogens. These are fever producing, originating outside the body, so bacterial surfaces. And endogenous, these are originated from inside the body by macrophages and peptides, right? These are like your macrophage something and your body's like, oh no, we got to release some pyrogens because we've got to get the fever up because we are under attack. So there's exogenous. These are bacterial and viral on the surface. And these are endogenous, which is fever-producing agents from within your own body. They raise your hypothalamus set point for body temperature. And your anterior hypothalamus neurons secrete prostaglandins, which also raise the set point. We talked about the set point and going back, and that's why you shiver, and that's why you're freezing when you have a fever, because your body wants to be at 102. Here's your stages of fever. You should have a normal body temperature. You get infected. Your hypothalamus resets the temperature. Body temperature rises. Now you have a fever, right? Body temperature oscillates around this new point of 101, 102. Infection hopefully ends. Your set point goes down to normal. You are going to be blazing hot. You're going to think you're so hot. This is when your fever breaks, and then your body's going to return to normal. Inflammation, yes, remember we talked about this. This is a local defense response. The purpose of it is to limit the spread of pathogens. It removes debris from the tissue. It's a sort of a cardinal sign of inflammation: redness, swelling, heat, and it causes a cytokine storm. That's what we need to talk about. Severe immune reaction. Body releases too many cytokines too quickly, and your body, this is one of the reasons why you have inflammation, because you have a cytokine storm. Also, it mobilizes the defenses. When you have any kind of inflammation, your body's like, dude, something's wrong. We got damaged tissue. We're releasing cytokines. What is the cytokines going to do? Remember, vasodilation. We're going to narrow everything down. Vasodilation from histamines. And you're going to deliver leukocytes and remove toxins and metabolic waste. You're going to vasoactivate chemicals. You're going to stimulate endothelial cells. You're going to increase capillary permeability. So your capillaries are going to become oozy and leaky. Why? Why would that be? Remember what we learned about the lymphatic system? Your capillaries are going to move more stuff into your lymphatic system, right? Your plasma proteins from the bloodstream are going to move into surrounding tissue. You're going to produce CAMs, which are cell adhesion molecules. These aid in leukocyte recruitment and diapedesis. The 5, you got heat, redness, swelling, pain, loss of function. Okay, again, neutrophil behavior. This is your diapedesis, phagocytosis. This is what is going to happen. Your bacteria, right? The first thing, you've got an inflammatory response that's going to release all kinds of things. You're going to increase the permeability of your capillaries. You're going to do some diapedesis. You're going to move all this out here. It's why you're going to be sort of oozy and edema, right? You're going to swell. There's going to be some chemotaxis, and ultimately that's going to release some phagocytosis that's going to eat those things. Okay, fibrogen, we should remember. Oh, we're at 15 minutes. Okay,